Bienvenidos al programa educativo Pediatras Expertos APLV.
Hello, my name is Professor Christine Lusher, and I am going to present to you today on immune dysregulation in allergy. This is my disclosure, and you can just read through that in your own time. So the objectives of the presentation are the following four. I'm going to first discuss what normal immune development looks like in infants. We're going to do a review of the dysregulation in the immune system that actually happens and then consequently results in an allergic reaction. So we're really interested in understanding what goes wrong in the immune system to drive an allergic reaction. We're going to then talk a little bit about how we manipulate the immune system to be able to overcome this. So if we understand so much about allergy, what can we do to be able to interfere with the immune system for benefit and actually prevent the allergic response? And then finally, I'll just briefly touch on the differences between breastfed and bottle-fed infants, specifically in relation to any factors that affect allergy. So let's start off with just looking at the normal immune system that happens in infants or develops in infants. If we look at babies when they are in utero, the mother's immune system is in a very different state to the normal state, and in fact becomes immunosuppressed. And the reason for that is that the mother's immune system is immunosuppressed in order for there to be no interference with the baby's growth as it develops in utero. So it becomes quite an immune privileged site. There's no interference and there's no negative impact of the mother's immune system on the baby. And I suppose in line with that, given that the baby develops in this type of immune environment, the baby's immune system when it's born reflects very, very well this type of environment. So if we just look at how the immune system firstly develops normally in utero, there's quite a big change between what happens in the first trimester, the second trimester, and then the third trimester, and into the process of birth. When we look in the first trimester, we really see a lot of progenitor cells beginning to be produced and being allowed to specialize or allowed to differentiate into other types of cells. So that as we move into the second trimester, you start to see the appearance of very specific immune cells. And these immune cells are ones that are going to be very, very important in terms of protection of the baby immediately after it's born. So, for example, you start to see macrophage appear in the intestine. The intestine houses about 70% of your immune system and therefore is one of the first sites that's populated with important immune cells. T cells, which we will talk about a lot in this presentation, start to appear in other sites that are important in the immune system. So the liver, the spleen, the thymus, and also in the intestine. And then the maternal antibodies start to cross the placenta, so the baby starts to have circulating antibodies, which again will be extremely protective once the baby is born. So at birth, there is a very big difference between very early in fetal development and then at birth. So there's a huge amount of the immune system has developed. You've got specialized cells ready to do their job. However, the immune system is very immature and has been developed in an environment where it's not been exposed to any pathogens, any antigens that aren't self, and the mother's immune status has actually protected that immune system as it's developed. The first thing that needs to happen after the baby's born is that immune system needs to be able to develop in a different environment where the antigens and pathogens that it's exposed to that may be harmful, the immune system needs to be able to respond to that. So at birth, you have fewer sentinel cells, which are there to be able to take part in surveillance to see if there's any pathogens present. These really important cells called dendritic cells, very, very low numbers. And the presence of these other cells, macrophage, although there are lots of them present, they are what we would term very immature. So their ability to be able to function fully is really, really hampered. So they don't have any experience at being exposed to pathogens. They're very poor at being able to recognize pathogens, and they're actually unable to be able to mount a proper immune response and function normally. So one function of these cells is the ability to engulf a pathogen and destroy it. And that's really important in terms of protection against infection. And these macrophages do that very, very poorly. The other thing that we see is we've got lots of T cells in the periphery, but these T cells have never been previously activated. They haven't been exposed to external antigen. And they're very, very inefficient in terms of their job. So they need to be developed and they need to be exercised in terms of how they need to respond. So there's a very immature immune system present in a newborn baby, and it is the key reason why newborn babies are so highly susceptible to infection, because their immune system has never encountered any single pathogen previously. So there's a huge challenge, therefore, for the newborn immune system. So the immune system has been largely suppressed because of the suppressive environment that it has developed in the uterus. It has no experience responding to pathogenic antigens. So we would term them to be very antigenically inexperienced. And actually what happens in the first few months of life is that the immune system becomes experienced by exposure to antigens and pathogens. So therefore, huge challenge for the immune system in a newborn is that there's a very high risk of infection. Now, there is a maternal, there's a level of maternal protection for these infants once they're born. So the passing of the antibodies through the placenta during development in utero, some of those antibodies will still be present for the first part of life. And then obviously the level of protection that's provided through things like breastfeeding, where there is a constant supply of those antibodies, but also immune cells and other factors. And we'll talk about that a little bit towards the end. I'll point a few of those out in the context of allergy. So there's a huge challenge for this very immature immune system in a newborn. And the end goal is really to be able to develop an immune system that has the ability to recognize and detect pathogens and mount an appropriate response for clearance. What's really important here to mention is that when any immune system comes in contact with a pathogen, the immune response is not the same. So the immune response to a virus is very different to the immune response to a bacteria, to that of a parasite, a yeast infection. So the immune system has a number of different types of responses. And when it encounters a pathogen, it switches on the most appropriate one to clear that pathogen. And that's where T cells come into play. And we'll talk a lot about T cells in the context of allergy. So one of the things that you want to do, because you've got these array of immune responses, they're all really orchestrated by T cells. So you've got different types of T cells. You really want to have a balance in the types of response that you mount. And this immune balance is really, really important for a fully functioning immune system. And I'll talk a little bit about balance and imbalance to show you where the dysregulation comes from in terms of allergy, particularly in relation to these T cells. So why are T cells important? So as I've already mentioned, when you have exposure to a pathogen, the type of immune response that you mount is very specific for that pathogen. Okay, and the really key cell in the middle of all of that, making all that happen, is a T cell. So if we just look at this slide here and we just see that the types of pathogens, so bacteria, viruses, helminths or parasites, different types of bacteria, intracellular, extracellular. So there's a huge array or class of pathogen that can actually cause an infection. And each of these presents themselves to the immune system. You have an antigen presenting cell that will be able to recognize a pathogen. And the job of that cell is to be able to present this information to a T cell and switch the T cell on. So for example, if you're exposed to certain bacteria and viruses, your pathogens are taken up by this antigen presenting cell and they switch on a particular T cell response. And you'll see here that this is a Th1 response. And then you have another type of response for a different pathogen, and that's called a Th2 response. We'll talk a lot about Th2 responses in this presentation because Th2 responses are very important in allergy. There's another type of T cell response called a Th17 response. And we also have regulatory cells, which are really, really important in maintaining immune balance and also in an infant becoming tolerant to non-harmful antigens as their immune system develops. So for every single class of pathogen, there is an appropriate T cell response. So there are a number of different options for the immune system when it comes in contact with a pathogen. And those options really are spread out into these 4 different subsets of these really important T helper cells. So T cells are really important. So if we look at the types of T cells that a newborn has and that of an adult, we will see a very big difference. So one of the things that I've mentioned already is this immune suppressive environment that the babies develop in. So when a baby is born, it has a predominance of this type of Th2 response, which you would normally associate with a suppressive response. It's a response that the immune system normally has to things like parasites and helminths. So it's definitely a very, very important immune response. The baby has a lot of these Th2 cells and much less of this Th1 cell. And this really makes a lot of sense because the mother's immunosuppressed and has this predominant Th2 response when she's pregnant. So the baby develops in that environment and has this predominant Th2 response. It doesn't have a very strong Th1 response. And that's because this response is really activated after you're exposed to bacterial pathogens, which the baby is not exposed to in utero in normal circumstances. Baby also has a lot of T regulatory cells, and these T regulatory cells are, while they're very important in immune balance, they're extremely important in tolerization of the baby to non-harmful pathogens once they're born. So you've got a lot of these T reg cells to make sure that the immune response doesn't get activated to the wrong things, so something that's not harmful, food antigens, milk proteins, for example, those kind of things. And as you see, there's a huge difference as we transition into looking at an adult immune system. And we see very much what you would like to see is this balance of a Th1 and Th2. So there's no dominance of Th1 or Th2. And this is what you would like in an optimal immune response from an adult. And it's what you're looking for in terms of an optimal immune development in an infant. The T regulatory cells are less. You don't need to be tolerized when you're an adult. That process has happened as you're an infant, but you still need T regulatory cells around because they're very important in switching off overactivated immune responses. So the balance is really important, okay? And there's lots of things that can affect that balance. So the important take-home message thus far is that when the baby's immune system develops, you want that immune system to develop to be able to have a very strong Th1 response and a strong Th2 response. So the ability to respond to pathogens is very, very efficient and it's very balanced. Exposure to different environmental factors can really influence this. So for example, one of the things that we saw in the last slide was that the Th1 response was quite poor in infants when they're born. So we need to exercise that immune response. We need to have that immune response gain experience so that you can mount a strong Th1 response when you come in contact with an antigen or a pathogen. And that Th1 response being exercised and being turned into a very efficient response, you need exposure to pathogens. So some of the things that we see, for example, in factors that favor that development of that Th1 response is situations where there is more exposure to pathogens. So presence of older siblings, exposure to daycare and creches, exposure to certain types of pathogens and infections very early on, and things like living in rural environments. So all of these are exposure to pathogens that are increased for these infants, and that allows them to be able to continually and consistently exercise their Th1 response, which is a really good thing for them to have done in terms of how they mount that immune response for the rest of their lives. Things that factor the Th2 response remaining dominant are things like widespread use of antibiotics, and that's hugely to do with the microbiota, which I'll mention a little bit later in this talk. Western lifestyles, diets, urban environments, sensitization to things like house dust mites. These things favour the Th2 response remaining dominant and this Th1 response not getting a chance to grow and develop. So this balance is really, really important. And the end goal is to have an immune system that doesn't have either one dominant, but rather has a balance between the two and is ready to mount the right immune response to the right pathogen. So balance is really, really important. So when we look at what happens when there is a dysregulation in any of these different types of T helper cell responses, this is what we see. Where we see a dysregulation in Th1 or Th17 responses, we predispose that individual to problems with inflammatory disorders. When there is a Th2 response that becomes dysregulated and overactivated, this results in an allergic response. And the T regulatory cells we always need for immune balance and for immune suppression. So the things that happen in a baby's immune system as it develops is really because of the experience that it has over time with antigens. And the exposure to those antigens, and lots of those are pathogenic antigens from bacteria and viruses, it allows the numbers of the different types of T helper cells to increase, and it allows the immune response to get a chance to exercise its effects in the situation where it's exposed to a bacteria or a virus. So exposure of infants to pathogens and them mounting an immune response early on in their immune development is a really important, and it's quite critical for full and optimal development of the immune system. The other thing that happens, as I've mentioned already, is that those T regulatory cells will decrease because we need those initially to be able to tolerize the infant against the antigens, so that it's able to tolerate the antigens that don't cause harm and is able to recognize those, particularly from pathogens, that do cause harm. The other thing is that the baby will increase its immune capacity. So we talked about macrophages. When the baby is born, they're very immature. Their ability to be able to recognize pathogens is poor. The ability to engulf them is poor. And a similar type of cell that's important in infection clearance is a neutrophil. So having macrophages and neutrophils with enhanced capacity, because the immune system is getting experience of exposure to these pathogens, is a really critical part of the immune development. And this tolerance that I've mentioned already is really important because the baby needs to be able to tolerate exposure to non-self antigens that are present in food. And that's why tolerization with the immune system in the gut is really important in normal development of the immune system. So there are lots of things that influence the immune system development in a baby. I'm focusing very much on the Th1 versus Th2 responses because Th2 is the immune response that becomes dysregulated in allergy. Huge part of this is controlled in the gut, where 70% of the immune system really exists, and therefore the gut microbiome is extremely important. This is heavily influenced by external factors, even as the baby is being born. So, for example, we now know from the literature that the mode of delivery, whether the baby comes down the vaginal canal or is born by cesarean section, there is a huge difference in the microbiota that then are laid down in the gut as a result of that. the antibiotic exposure. So antibiotics destroy certain classes of microbe in the gut and therefore allow different microbes to be able to grow. So exposure to antibiotics very early on changes the gut microbiome. Whether a baby is breast or bottle fed, and again, I'll mention this towards the end, huge impacts on the microbiome. And then other factors, for example, we've seen a huge body of literature, particularly over the last decade, where the maternal nutritional status and exposure to certain types of foods during pregnancy has a lasting effect on the infant, particularly in terms of the microbiota. So the microbiome is really important in this whole development of the immune system. It influences the Th1, Th2. And while we won't go into a huge amount of detail about the exact mechanisms that are involved, I just think it's important at this stage that when we are trying to understand about the Th2 response being dysregulated in allergy and why that's not a normal development of the immune system in an infant, we need to understand that there's a huge influence from the microbiome in this process. So if we want to understand allergy, we need to understand why this Th2 response is dysregulated. So if we just look briefly, let's take a couple of moments to look at allergy, to look at atopic disease, to look at the kinds of diseases it covers, and then talk a little bit about food allergy. So we just get a bit of a context for what's happening in infants at the moment. About 20 to 30% of children are affected by atopic disease. This is a huge number. It is made up of the increasing incidence in a variety of atopic diseases, including things like asthma and eczema and food allergies. And all of these things have increased over the last number of decades. And if we just refer back to one of the previous slides where I said that a lot of the factors that really influence the immune system development in a baby are external factors, it is actually these external factors, environmental exposures, use of antibiotics, change in microbiota, that is really driving this huge increase that we've seen in atopic disease. Asthma, food allergies, eczema, they are all on the rise, and they have been for the last number of decades. So we have all these infants whose immune systems have not developed properly, and they therefore have this predisposition to atopic disease. So food allergy arises when the baby is exposed to a particular food antigen, and the immune system, instead of becoming tolerant to that antigen by virtue of the presence of these T regulatory cells, for example, that's really important, as is the microbiota of the gut, there is a breakdown in the tolerance mechanisms that normally happens. And the immune system is allowed to respond to these. And when it responds in a way that drives an allergic reaction. Okay. If we look, for example, at children with food allergies, particularly those like cow's milk protein, hen egg allergies, and wheat allergies, in about 50% of children, they will resolve by themselves in the first 5 to 10 years. And what happens in those situations is that where there is an initial allergic reaction to a particular antigen from one of these proteins, that over time, the immune system learns how to tolerate these antigens, and the tolerance mechanism kicks in, but it kicks in rather late. One of the things that happens, though, in these infants, as we'll see in the next point, is that they become they've become atopic. So there is an issue with the way their immune system has developed. And while they might resolve an allergy to cow's milk protein, for example, they may develop eczema, they may develop atopic dermatitis, they may develop asthma as another allergy, because their immune system is atopic. There is a dysregulation of Th2 already. So even though when the immune system develops and there is an allergic reaction very early on, these children, even if it's resolved, are high risk for developing other types of allergies. So the issue that happens very early on in those immune systems has a lifelong effect. There is a lifelong impact when this immune system does not develop properly, optimally, and in a balanced way very, very early on in life. So I thought I would just put this up because it is I suppose, a really good way of being able to, I suppose, emphasize the point I've just made, which is that if you have issues with the immune system development early on, they start down this road of what we call this atopic march, which is that the issues that arise because of these immune system dysregulation, they lead on to one thing, they leave the child susceptible to something else, and it really just starts to roll along. And if we just look at things that happen in the gut microbiome that influence dysregulation of the immune system development, something that happens when the immune system has an allergic response, for example, you get changes in the barrier function in the gut, you get breakdown in the gut, you get leaky gut. Those kind of things, while they present as a food allergy, they then leave that individual with issues in terms of gut membrane that leaves them susceptible to things like IBD, gut dysfunction much later on in life. And we know now that there's a huge relationship between the gut and the brain, and therefore even things like neuropsychiatric disorders can result. All of these things are influenced by environmental factors, the genetic background, for example, and even epigenetics. So the point I want to make with this particular diagram is that any dysregulation very early on in the immune system will have consequences and leave individuals susceptible to other diseases and other manifestations of immune system dysregulation for the entirety of their life. And this is obviously very heavily influenced by external factors. And what's really important there is that I think it really emphasizes that optimal immune development very, very early on is important for life, not just for that allergic reaction that you might be getting as an infant, or some deficit in the immune response very early on in an infant. It lays down the foundations of the immune system for life. So it's really, really important that we understand the immune system development in an infant and understand how we avoid, at any cost, the dysregulation of that immune system early on. There's really very little chance to be able to fix it permanently. So infants who develop atopic diseases and allergies have this dominant Th2 response. Okay, they've got a lack of tolerance, they don't tolerate antigens well, they don't exercise the immune system to be able to tolerate them. They're allowed to react to them, and they're allowed to mount an immune response to them. And really, in these individuals, they're mounting an immune response to something that's not harmful to them. Okay, so you've got an immune response that's out of control, and allergic reactions are exactly that. The allergen, okay, or the antigen that the immune system is seeing as a pathogen or as something that's dangerous, when it is presented and taken up by the antigen presenting cell, it activates these Th2 cells that we've spoken a lot about. These Th2 cells produce lots of these tiny molecules, messenger molecules called cytokines, and they are able to drive B cells to make antibody. And the particular type of antibody that they drive is IgE. And IgE binds to mast cells and instructs them to degranulate. And when mast cells degranulate, they release histamine. And most of us will have heard of histamine because if you have an allergic reaction, one of the first things that you take is an antihistamine to block the effects of histamine. So anytime a child has an atopic disorder and reacts incorrectly to these harmful allergens, this is the process that gets switched on. And the symptoms that they exert because of this allergic reaction are really very much mediated by this massive production of histamine. And again, just to emphasize that, of course, the microbiome plays quite a significant role, and we know that there are lots of factors that really influence this. And one of the things that I really wanted to point out here is that if we look at a child who has a food allergy and we look at a child that is otherwise healthy in terms of its immune system, there is a big difference between their microbiota and a big difference between the environmental factors that they have been exposed to very early on in life. So there is an opportunity when an infant is born with a brand new immune system that is inexperienced, that wants to develop to be balanced. There's a huge opportunity to be able to make the right choices and expose those infants to the right things to be able to get optimal development of their immune system. So there's this very short window as an infant for getting the immune system right for the rest of your life. It's hugely influenced by the microbiota. So if we just take one example of cow's milk protein allergy, which is one of the common allergies that infants can develop very early on in life, what happens in that response? So here what we would see is we see milk proteins that have been broken down by the digestive system. And those milk proteins should not be recognized by the immune system. In fact, the immune system, when it recognizes them, it should be tolerized so that it doesn't respond to them. But actually the exact opposite happens. So the antigen presenting cell picks up these little antigens from the milk that have been made from the milk protein. It switches on these Th2 cells that I've spoken a lot about. They make these cytokines. The cytokines drive the B cells. The B cells make the antibodies, and the antibodies instruct the mast cells to make all this histamine. So this is a very simplified diagram that just shows you that this is how a Th2 response gets switched on in the absence of a harmful substance. And it's to do with the fact that the infant has not tolerized itself to be able to be tolerant to these food antigens. The current approach for lots of different reasons is if there is an antigen, such as a cow's milk protein antigen, that causes an allergic reaction in a child, that the current approach is complete avoidance. Okay, and the way that that avoidance happens but also being able to provide a cow's milk protein to a child as a form of nourishment is that they are extensively hydrolyzed. So when a milk protein is digested, it's digested into a number of smaller fragments. Okay, and those smaller fragments are recognized by the immune system. So those small antigens that come from this main protein are recognized by the immune system, and they drive this allergic reaction. The way around that is to hydrolyze the milk protein before the infant takes it and digests it, and to extensively hydrolyze it so much that even the individual antigens that come out of that hydrolyzation process can't be recognized by the immune system. So in other words, you hydrolyze the protein so much that even the individual antigens are not recognized at all. And therefore, the infant is able to tolerate those hydrolyzed proteins and doesn't have an allergic reaction. So you can still give them cow's milk protein in a different format for nutritional value, but you are not activating the immune system to drive this allergic response. And this is one of the main ways in which cow's milk protein allergy is dealt with. But we know a lot about the immune system. So we know that an individual who has a cow's milk protein allergy, for example, we know that they don't have immune balance. We know that they have this really strong Th2 response to the milk protein. We know that their microbiome has changed in some way. So if we know so much about what's wrong with the immune system and why it reacts in this way, then maybe we should be looking at other ways of using this information and manipulating the immune system to be able to overcome this dysregulation. So can we dampen down or suppress this Th2 response? Can we change the microbiome? Can we restore balance? So these are all really important questions in terms of coming up with alternative strategies to be able to manage this dysregulation, and particularly in infants. So with that in mind, I'm going to give you one example, and then I'm going to present some of my own research, which gives a lovely exemplar of what is possible if we manipulate the immune response and try and switch off this Th2 response. So this example here involves a peanut allergy model, and it's a peanut allergy model in a mouse. And essentially, once this mouse is exposed to peanut antigens, it mounts this really strong Th2 response, and you get a full allergic reaction. Now, one of the things that's really important in balance is that there's a relationship between Th1 and Th2. And if you have an activation of a strong Th1 response, when you have a Th2 response present, the Th1 response can actually suppress the Th2 response, and you can actually restore balance. So in this situation, what happens is we add in, when we expose the mice to the peanut allergy, we add in something else that will drive this Th1 response. So one of the things that drives the Th1 response is this cytokine called interleukin-12. And what you do is you cleverly put these interleukin-12 cytokines into little nanoparticles, and that allows you to deliver them inside into the mouse. So when you give it the peanut antigens, and then you give it these IL-12, you don't have a chance to activate this really strong Th2 response. In fact, you mount a Th1 response, and you don't get the allergy. You don't get the Th2 cells driving the B cells to make the antibody. You don't get the antibody asking the mast cells to degranulate, and therefore you don't get an allergic reaction. So this is a really good example of manipulating the immune response so that when it goes to switch on this Th2, you've put in something else that will skew the immune response the other direction, and instead will mount this immune response, this Th1 response, and you will have this immune balance. So it's a really good example of the kinds of things that you can do to be able to manipulate the immune response. It's very responsive to factors like cytokines, for example, and you can totally change an immune response in an animal by just administering cytokines. So we know so much about the immune system. There's so much more that we can do. So if we know how the immune response becomes very dysregulated in a cow's milk protein allergy, for example, one of the things that we've talked about a lot is the fact that we drive a very strong Th2 response. Now, these Th2 cells make antibodies, interleukin-4 and interleukin-13. These are really important cytokines, not antibodies, sorry, cytokines. They make these cytokines, and these cytokines are really important for instructing that B cell to make the antibodies. So if we want to manipulate the immune system and switch off this allergic response, instead of trying to deal with why the immune system is not tolerized to these, why don't we interfere here, and why don't we try and manipulate the immune system to stop this Th2 response from becoming switched on? Okay, so one of the things that we've been working on in my research group is looking at milk hydrolysates. So these are hydrolyzed milk proteins, which give rise to a vast array of hydrolysates. And we've become very interested in looking at how these hydrolysates instruct Th1, Th2, and Th17 cells. And what we've been able to do after we've done a huge amount of screening of these and looked at what they do in vitro in our cell models to particularly Th2 cells, I'm going to show you some data where we took one of these hydrolysates that gave us very, very good responses by suppressing Th2 in cell models. We took it into an animal model of allergy to see if it could, in fact, suppress a Th2 response in that model, but also could it suppress the allergic response and the symptoms in that animal. So we chose a very simple ova model. So the mice are sensitized to the ova over time. And then essentially, when you wait a period of time and you challenge the mouse with the ova protein, it mounts an allergic reaction. Okay, so it reacts very, very strongly to it. So we used this model. We used one of these hydrolysates that we'd had some really nice data from. For the purpose of this talk, we'll just call it 147. Okay, so this 147 hydrolysate is a very simple hydrolysate. It's a breakdown product of a cow's milk protein. So the mice were sensitized to the ova. They were given alum as an adjuvant. And we then gave them the milk hydrolysate over the period of several days. We gave this by gastral gavage. So essentially, they were fed and exposed to this milk hydrolysate in the gut that we were very much hoping could manipulate its T-cell responses. And then all the mice were exposed to the ova protein. And we expected in the mice that were our positive control mice, we expect them to have a very full-blown allergic reaction. So let's look at some of the data. So if we look at some of the data, one of the things that we always measure is something that we call this disease activity index score. And this score really takes into consideration lots of indicators of disease from the animals themselves. And one of the things that you will see is that the mice that are oversensitized, which are having the allergic response, every time that they are challenged with ova, they have this spike in the disease activity because they are having an allergic response. And when you gave these mice this 1,4,7 hydrolysate that performed very well in our cell models, you can see really that the red just stays at the baseline. They do not have or display any of these symptoms of the disease activity, and they show no allergic symptoms whatsoever to the ova. So they should be responding to the ova, but we've given them the hydrolysate and now they're not responding at all. So we wanted to have a closer look and see what was happening in the gut and what was happening with its Th2 cells. So firstly, let's look in the gut. If you remember from that pyramid that I showed you, the consequences of changes in microbiota, the overactivation of and the allergic responses that take place in the gut, they disturb the intestinal barrier, they disturb barrier integrity. These kind of gut problems lead on to other problems later in life. So it's really important that we look in the gut during an allergic reaction and see what happens, and then look at what happened with our 1,4,7 hydrolysate to see if it actually gave any benefit. So this is a cross-section, some histology from the gut. You see in the normal animals, we see these lovely crypts, which is very typical of what we see in the intestine. And we see this beautiful architecture with the membrane integrity in place. These are the oversensitized mice. So these are the ones that were exposed to the ova and had an allergic reaction. Their disease activity score was high. The gut architecture has been destroyed. Their barrier function has been impacted. And then if we look at these beautiful slides with the 1,4,7 hydrolysate that we gave to these animals before we gave them the ova, which should have called the allergic reaction, and we see beautiful architecture in the gut. The crypts are all intact and there's no issue with the membrane integrity. So when we've given this 1,4,7, we have stopped this allergic reaction because we understand a lot about what the immune system does when it has an allergic reaction, and we put something in there that we think blocks that. So the most important thing that we look at then is we look at the Th2 response. So what actually happened in the gut? So we take out the gut, we look at the cells, we look at the responses. What happens to Th2 cells? And in order to be able for us to tell that, we actually looked at the cytokines that the Th2 cells produce. So they produce lots of IL-4 and lots of IL-13. And if you look at the oversensitized mice, where you get lots of this because they're having a big allergic reaction, and this is the group that got the 1,4,7. This is the IL-13 oversensitized mice having a big reaction here, and it's less with the 1,4,7. So the cytokines being produced by Th2 cells that are really important in driving the B cells to make all that antibody for the mast cells, those cytokines are down, the Th2 cells are suppressed. And then interestingly, T regulatory cells produce this important cytokine called TGF-beta. It's really important for tolerance. It's really important for balance. And actually what you see in the mice that get the 1,4,7 is that they've got increased amounts of TGF-beta, almost like a body's attempt to be able to tolerize and to be able to balance the immune system and not have this allergic reaction. The presence of TGF-beta is a really, really positive thing to have around if you're trying to prevent these type of allergy responses. I'll refer to TGF-beta again in a few moments when we actually look at what happens look at some differences between breastfed and bottle-fed infants. So the other thing that happens is that one of the things that we find in the circulation when we have a strong immune response, particularly a strong allergic response, is you get this subclass of IgG, you get IgG1 being produced. When we look at the oversensitized mice, we see lots of this IgG1 in the circulation. And with 147, we see that drastically reduced. So all of the indicators of allergy, Th2 cells, barrier function, architecture in the gut, IgG in the circulation, they are all decreased. And more importantly, the disease activity score for these animals is practically the same as the control animals. They do not get an immune response. And that's happened because we have manipulated the immune system not to be mounting this Th2 response. So this 147 prevents the clinical symptoms observed in these oversensitive mice. They resolve this superficial inflammation and breakdown of the tissue in the gut. They attenuate or block this Th2 dominant response in these mice, which is exactly what you want to try and block this allergic reaction. They drive these T regulatory cells, you get lots of TGF-beta, and they drive down this IgG1 antibody, which is what you make when you have an allergic reaction, and it's very much seen in the circulation. So all of these would indicate that we have the ability to be able to suppress Th2 responses and then potentially be able to suppress allergic responses in an infant to a particular antigen. I think the cow's milk protein antigen is a really good example of that. So just finally, one of the things that I just wanted to touch on, and I know that in the other presentations in this series, this will be discussed in more detail, but I just wanted to touch on, because we're interested in immune balance and we're interested in Th1 versus Th2 and the regulation of the tolerance, I just wanted to touch briefly on a couple of key differences really between breastfed and bottle-fed infants. One of the things I'm not going to go into, but I wanted to really highlight is that there's a very big difference in the microbiota. So the percentages of the different classes of microbiota found in the gut of these infants is drastically different, okay? And this is really important because it means that not only is the gut microbiota different, but the way in which their immune system in their gut is developing is also drastically different. One of the things that we see in breastfed babies is that they have higher levels of TGF-beta. Now, I mentioned TGF-beta a couple of moments ago when I showed you some of our data from our research. TGF-beta is what's produced by T regulatory cells. It can drive T regulatory cells. They're really important in tolerance. So breastfed babies have lots of TGF-beta. They're tolerized. Their gut has the ability to tolerize them to antigens, and this promotes a more balanced immune system development. So this TGF-beta is very important in this immune balance. The other thing it does is that it limits IgE production. And IgE production is what's driving the mast cells to produce all the histamine in the allergic response. So if TGF-beta can limit that, it's almost something that's trying to block an allergic reaction from actually taking place. And as I've mentioned already, the microbiome is very, very different. And if we finally just look at a table where we look at individual components that are transferred from the mother to the baby during breastfeeding, we see lots of components that I've mentioned throughout my talk. So I think it's just important to be able to point some of those out. We've mentioned the microbiota. It is very, very different in terms of what breastfed babies are exposed to in terms of the microbiota in the milk. And that's really, really important because that microbiota has a direct route to the baby's gut and starts to populate the baby's gut with different classes of microbiota for optimal development of the microbiome and the immune system. There's a number of cellular components in there. There are high numbers of things like macrophages, T cells, neutrophils, all being transferred to the baby very, very early on in life. So the immune system is getting this a massive boost of even the immune cells, not just what we would have always kind of taught about breast milk, which is just about the antibody and transfer from mother to baby, but all of these immune cells and all these microbes are also being transferred. And then when we look, obviously, the big one is the antibodies. Okay, this passive transfer of antibodies provides huge protection for the baby very, very early on in life, and is almost like the first line of defense while the baby's immune system is developing its own line of defense. So oligosaccharides, we'll have heard a lot about those in this lecture series, extremely important. So this is just to point out that immune system components are transferred from mother to baby and have an important role to play in the immune system development. So I hope that the take-home messages from this presentation are very clear. So just to finish up, I'd like to really emphasize those, that the development of allergy in infants is because of this lack of tolerance, this lack of balance, and this dominant Th2 response that gets a chance to take a foothold very early on in the immune response, because this Th1 is not being exercised enough, because this balance is not developing, this Th1 Th2 balance. All of this is heavily influenced by the microbiome. Everything that's happening here is happening in the gut. It's really, really important. And then finally, I think a really important message is that we can target the immune system and manipulate these allergic responses by using external factors. So I've just given you the really good example of using these hydrolysates, for example. So things that are safe, that are able to be ingested, and that manipulate the immune response and really influence the immune response in a baby when it's exposed to an antigen. Also, because we know so much about the microbiome, anything that manipulates the microbiome and changes it in a positive way is also has potential to be able to overcome this dysregulation that we see in infants. So I hope you have enjoyed my talk. I hope that the take-home messages are very clear from the information that we've given, and I look forward to another lecture series with you soon. Thank you very much.
Hemos finalizado con la sesión del día de hoy. Cualquier consulta pueden escribir al número que se encuentra en pantalla. Hasta pronto.